So, good day everyone. So, we are the group 4 and we're going to present our very own do-it-yourself genetic modified organism, or the crop that we're going to use is the grapes, or also known as Vitis vinifera. So, our topic is all about how to alter the genetic input of a grapes and how to they become resistant to any adversities, even in the unfavorable conditions in their development. So the reason why we pick Vitis vinifera, or also known as the grape, it's because it is one of the most economically significant fruit crops. This is the crop that used for making wines, raisins, and also table grapes, which is the we used to eat the fruit. So the problem of this is some varieties of Vitis vinifera are highly susceptible to various pathogens, such as in the dimension of Pierce's diseases. So, our goal here is to transfer natural resistance from a trade of from wild grapes to commercial varieties that using accessible to biotechnology. So, in a valuable but vulnerable crop, it needs to be protected, and this project is aims to provide the protection by moving a gene from a natural resistance from a resistance of a wild relative into a commercial varieties for using genetic engineering.
Good day everyone. I am Angel Arsumbilan and today I will be discussing persist disease, a serious threat to grapevines. Persist disease or PD is caused by a bacterium called Xylella fastidiosa. This bacterium blocks the plant's xylem, which is responsible for transporting water. As the xylem becomes clogged, the vine slowly dries out and eventually dies. One of the biggest reasons PD spreads so quickly is because of its insect vector, the glassy wing sharpshooter. This insect carries the bacteria and transfers it rapidly from one vine to another, causing widespread infection. The symptoms can be seen on the leaves, which show yellowing and browning around the edges. Next is the genetic solution. So because persist disease causes severe damage, scientists are looking for long-term solutions. Traditional breeding for disease-resistant grapes takes many years, sometimes even decades. Another problem with traditional breeding is what we call linkage drag, which means the quality of the grape, especially its flavor, may be negatively affected. Genetic engineering gives us a faster and more precise solution. Instead of changing many traits, scientists can insert just one resistance gene into the grape. This helps protect the vine from persist disease while keeping the original flavor and quality of the grapes. This is why genetic engineering is seen as promising solution for managing persist disease.
About PDR1. PDR1 is a disease resistance gene found in a wild grape species, Vitis arizonica, that help plant recognize and defend against persist disease. Vitis arizonica, a wild grape native to North America, naturally evolved resistance to persist disease. Persist disease is a deadly condition caused by the bacterium Xylella fastidiosa. PDR1 was identified through screening of wild grape population for natural resistant traits. PDR1 is a gene type that encodes a receptor-like protein that plays a role in plants' immune system. It detects molecular signal from Xylella fastidiosa, and upon recognition, it triggers a self-defense that limit bacterial spread and damage. For its mechanism, the PDR1 gene functions as a molecular sentinel within a grape cell, enabling early detection and defense against Xylella fastidiosa, the bacterium responsible for Pierce's disease. When this pathogen enters the plant vascular system, PDR1 encodes a receptor-like protein that recognizes specific bacteria signals often referred to as a pathogen associated molecular pattern or PAMP. Upon recognition, the receptor activates a cascade of intercellular signaling events, including calcium flux and kinesis activation. This leads to the production of antibacterial compounds, reinforcement of the cell wall, and in some cases, localize the cell death and contains the infection, a process known as the hypersensitive response. By triggering the defense mechanism early, PDR1 helps prevent systematic infection and contributes to the plant's overall resistance. Because PDR1 originates from a wild grape species closely related to cultivated varieties, it used genetic improvement and considered cisgenesis genesis preventing species compatibility while enhancing disease resilience. Here is the step-by-step in acquiring desirable agrobacterium. First, identify and prepare the T plasmid. Agrobacterium tumefaciens naturally contains a T or tumor-inducing plasmid. Scientists removed the disease-causing genes from a plasmid to disarm the bacterium. Next is insert the gene of interest. The T plasmid is cut using a restricted enzyme. A foreign gene, PDR1 for disease resistance, is inserted into the T plasmid region of the plasmid. This creates a recombinant T plasmid. Next is reintroduce the recombinant plasmid into agrobacterium. The engineered plasmid is reinserted into agrobacterium, which now acts as a delivery tool. Next is infect the plant cell. A suspension of engineered agrobacterium is used to infect the plant tissue, like grape leaf disc. The bacterium transferred the T plasmid, which inserted the gene into the plant cell genome. Fifth is culture transformed cell. Infected plant cells are cultured in vitro under controlled condition. Only cells that successfully integrate the gene are selected and grown. Sixth is generate transgenic plant. This cultured cell develop into a whole plant. The resulting plants are transgenic, expressing the inserted gene or resistant to disease.
And now let us move on to or talk about plasmic construct design. But first, let us understand what is plasmic construct design. Well, it is a process of creating a circular piece of DNA with specific functional elements to achieve a desired biological outcome, such as producing a particular protein. Well, it involves assembling a gene of interest with other components like a promoter, which initiates transcription, and a terminator sequence to stop it. The genetic payload. Well, a binary plasmid vector is engineered with the following key components, and this includes promoter, gene of interest, selectable marker. In a promoter, it acts as an on switch to ensure the gene is always active, and in gene of interest, the DNA sequence coding for resistance. And lastly, the selectable marker, it confers antibiotic resistance, allowing for the identification of successful transformed cells.
So this is Karen Spredo and I will be discussing to you the somatic embryogenesis. So it is the process by which they are producing genetically identical plants through a tissue culture, and it involves the transformation of somatic or non-reproductive cells into embryos, which later then develop into a whole plant. And um, in the process, its key steps are first the induction. It is the inducing of callus growth from an anther or ovaries. And next is the co-cultivation. It is the incubating of callus with engineered agrobacterium. Third one is the selection. It is the growing of tissue in an antibiotic media to select transformed cells. And lastly, the regeneration. Um, it is the triggering the formation of shoots and roots from a surviving embryo, and this process is particularly relevant to woody plants like grapevine as it rapidly increase the production of genetically modified or impaired varieties.
After the transformation process, we expect three key outcomes. First, PCR testing should confirm that the PDR1 resistance gene is successfully integrated into the grapevine genome. Second, when exposed to Salmonella fastidiosa, the engineered plant should show strong resistance, meaning little to no leaf scorching, dieback, or other typical Pierce disease symptoms. Finally, because the PDR1 gene comes from a related grape species, we expect fruit quality to remain unchanged. The berries should keep the same flavor, sugar levels, and overall characteristics as the original variety. Overall, the goal is a vine that resists disease without sacrificing the trait that make Vitis vinifera valuable.
Since this project involves working with GMOs, it is important to follow strict DIY and safety guidelines, especially in a student or community laboratory settings. First, we need to remember that Agrobacterium tumefaciens is considered a plant pest. Even though we use a disalarm version, proper disposal is required. All culture should be autoclaved before disposal to prevent accidental release in nearby plants. Next is any genetically modified grape plant must never be released in the environment. This plant must remain in controlled laboratory or greenhouse condition and follow biosafety level 1 or 2 protocols. We must also follow government regulation regarding GMO handling and research. Field trials of genetically modified crop are strictly regulated, and authorized release can have legal consequence. Finally, ethical practice should be observed, such as transparency about GMO works and carefully consideration with using antibiotics resistant makers. Professional research, these markers are being replaced to prevent potential environmental impact, and these guidelines ensure that the project is considered safely, responsibly, and ethically. On this slide, we present the expected result of the genetically modified grapevine carrying the PDR1 resistant gene. First, for the genotype, we expect the modified plant to test positive for the PDR1 gene through PCR analysis. This confirmed that the resistant gene has been successfully inserted into the grapevine genome. Next, for the phenotype, we anticipate that when the transformed plant are exposed to Salmonella fastidiosa, they will show little to no symptoms of Pierce disease. This means that there should be no leaf scorching, no dieback, and overall healthier vine compared to the susceptible varieties. Lastly, in terms of fruit quality, we expect that important traits such as sweetness, acidity, and overall flavor will remain identical to the original. This is because we are inserting only the resistant gene and no flavored gene related are altered. Overall, this result will show that the PDR1 gene provides strong disease resistance without compromising fruit quality.